in a ligand dependent fashion. So this ligand is there. So you can see the way RNA is folding here has changed totally. Earlier ribosome binding site was free, so the translation was occurring. Okay, but if the ligand is there, then what can happen? This is one situation where RBS gets sequestered and then ribosome binding site is hidden. So please remember this diagram. This is going to be central to your understanding of riboswitches or explaining the way riboswitches act. Now, riboswitches are comprised of an aptamer domain. I had told you last time. Let me go a few slides back if I can find that slide for you. Now, this one. Okay, so this is the aptamer domain. Aptamer domain is where the molecule will bind. You can see here, this one, the molecule has bound here. Now you can see these two sequences, they were away. But once this molecule is bound, they have come together and now they are doing like this. And that is where now in this case, actually here, so ribosome binding site was free, and once the molecule is bound, then it gets occupied. So I am giving you a more complex example here. So aptamer binding, wherever the binding is there, it is called as aptamer, and wherever this regulatory thing is there, whether it is transcriptional regulation or it is translational regulation. In case of translational regulation, RBS will play a role, whether it is bound, sequestered, or it gets free after the binding of the small molecule, depending on case to case. So different riboswitches have got different kinds of mechanism. So I hope this much all of you have understood, and then now we can move on further. So riboswitches examples are like, there is a riboswitch for B12, coenzyme B12. There is a riboswitch for S-adenosine methionine. There is a riboswitch for flavin mononucleotide. Purine riboswitch is there, where the nucleotide can bind and regulate the gene expression. Amino acid based riboswitch is also there, lysine riboswitch. Then this is GLMS
सर
riboswitch. Yeah,
सर स्लाइड चेंज नहीं हो रही.
नहीं हो रही है. Okay, so I'll have to stop sharing and then start it again. Okay.
Yes, sir.
भाई जो लेट आया ना, they should come in time. Now it disturbs the whole rhythm of the class. Okay. पटेल साहब भी आ गए. Patel, welcome. Okay, now let us start the share. Can you see the slide?
Yes, sir.
But now the slide is not moving. अरे यार क्या है ये? Yeah. Now the slides are moving. Okay, thank you, Aryan. So moving on to the next slide, next point, riboswitches are composed of an aptamer domain that I had shown you, which recognizes the ligand, where the ligand binds or the small molecule binds, and an expression platform which couples ligand binding to change in gene expression. So this is where is the expression domain, and this is your aptamer domain. Okay. Now going on to the next. Oh, are you able to see now?
Yes, sir.
Yeah, you are able to see, but slides are not moving. What could be the problem? Can anyone suggest me? It never used to happen earlier. Should I share the entire screen? Will it help? But Aryan, it's not moving, man.
सर एक बार मीटिंग लीव करके ज्वाइन करके देखो
हाँ प्रत्युष,
सर एक बार मीटिंग लीव करके ज्वाइन करिए सर मे बी सम टाइम इशू होता है
मैंने लीव कर दी तो ज्वाइन कैसे करूंगा यार? मैं तो स्टार्ट किया, मैं हूँ होस्ट हूँ.
सर वो चेंज हो जाता है
अच्छा एक बार और ट्राई करते हैं. विंडो, पावर पॉइंट स्लाइड शो.
ये
ओके,
सर हो गया सर
ऑलराइट, ठीक है. अभी ये देखिए, सो नाउ वी आर गेटिंग इनटू सिंथेटिक बायोलॉजी. अभी तक वट एवर वी वर स्टडिंग फॉर राइबोस्विचेस, it was about the natural riboswitches which are occurring in nature. Now we can even design riboswitches using synthetic biology, and we can make really cool biosensors using such riboswitches. Okay, so uska basis is same. Like I had told you that after the riboswitch you can add a reporter protein which can be a green fluorescent protein. So this is what people have done, and I'm going to show you two kinds of synthetic biology approaches for riboswitch design. One of them is which is inspired by the natural riboswitch which are present. So you can see here, so this is a riboswitch which is based on theophylline. Theophylline is a molecule which looks quite a lot like caffeine molecule. Okay, caffeine के जैसे ही theophylline molecule है. And then there is a riboswitch for theophylline because theophylline is a natural molecule. And then this theophylline binds to this part here. I don't know whether I can you see the cursor? Are you guys able to see the cursor? Mouse cursor? Nahi? Okay, so if you look at the panel A in the left, this panel shows theophylline written, and then there is an aptamer domain which is green color. Now you can read at the bottom, sequence of a portion of this 5' region of a parent synthetic riboswitch with 8 bases is green in color. So the green color part is the aptamer domain, and where this small molecule theophylline binds, maroon color ka jo molecule hai, it binds there. And now at the bottom, if you see on the right side, there is an AUG start codon which is peach in color, and the ribosome binding site is pink in color, and you can see that it is written over there. Now what people have done is, this aptamer, this riboswitch was not very efficient. So what people have done is, now go to the C panel. You can see in the C panel, it says at the bottom, rather than parent, it says randomized. So what they have done is, they have randomized this region. So what is the randomization? It means in parent molecule, if you see in the left, the first blue highlighted nucleotide is U, and it goes like U, A, U, A, G, right? So in this case, if you see, it is N, and then the ribosome binding site, and then the start codon is there. So in this study, what they did was, they changed these UAG into randomized sequences. So which means, instead of U, they made a variant where it could be ATGC, not T but AUGC, and the second nucleotide also AUGC. So you will have combination of all these molecules. So on the top, if you see in the blue part in C panel, there is a big circle which says N4, N5, N6, N7, and 8. So it shows if there are randomization in 4 nucleotide, then you will have 256 combinations. If you have 5, you will have 1024, and so on. So more nucleotides you randomize, more variants of riboswitches you will get. Now the question is, how do you select the best one? So you select the best one because you have added, after the start codon, you have added a green fluorescent protein at the end. Now let me see. And it got stuck again. Oh man, I don't like this. So after AUG, you will have the green fluorescent protein, and you will start measuring up for all the variants. You will have individual clones, and then you start measuring the fluorescence of those individual clones. Now, the more brighter a clone is after adding theophylline, better riboswitch it is. So that is how this study actually found that lot of riboswitches now can be designed based on similar properties. Okay, so now if you see here part A, okay, part A on top. Can you see this slide?
Yes, sir.
Okay, yeah, thank you. So you see here the yellowish color, orangish color molecule is binding here, and this reporter is there which is producing a signal, this green signal, A panel only at the top, left top. Okay, so similar approach was used by these guys, and then they selected one which was very efficient as compared to the parent. So left side wale ki comparison mein C1 they selected, and they found that it is really very efficient. So that is one way synthetic biology is making really good biosensors also. Okay, now I just want to take you to a totally, part of synthetic biology only, but how we can select riboswitches, new riboswitches. In this case, the riboswitch was already there in nature, but you can make a totally synthetic riboswitch which doesn't occur in nature using some approach which is like this. So first, rather than RNA, I'm going to tell you about DNA. And when we say DNA, making an RNA is very easy. Can anyone tell me how we can make RNA from a DNA in a test tube? Anyone? Any guess? Shilpesh?
Yes,
Shilpesh, are you there?
yes, sir.
तुम पिछली कुछ क्लासेस में गायब कहाँ हो?
Sir, I was there. Uh,
No?
in offline class, I was not.
Why not? Online class तो भाई exception है.
Yes,
Why you
sir.
are not interested?
Sir, I'm interested, sir.
So
Sir,
next class में आपको
I
you
was
have
not
to be there.
there.
Okay? Okay, so बताओ अभी मुझे. शिल्पेश,
Yes, sir.
डीएनए से आरएनए बनाना है तो क्या करना होगा?
So we can do simple translation to
Translation? प्रत्युष? प्रत्युष मंगल?
transcription, sir.
Yes, sir.
प्रत्युष, डीएनए से आरएनए हमने बनाना है टेस्ट ट्यूब में, कैसे बन सकता है? अभी तुम जो है ना गूगल ना करने लग जाओ.
हाँ सर.
अच्छा, डीएनए से आरएनए बनता कैसे है?
सर ट्रांसक्रिप्शन.
ट्रांसक्रिप्शन से. So can you do a transcription in the test tube?
मे बी सर.
Okay, so what will you need for that?
Required enzymes for that to perform that.
Okay. Yeah. Which enzymes?
Tag
Okay.
polymerase.
Tag polymerase क्यों? Harshita, are you there? Harshita Verma.
Yes, sir.
Okay, tell me. Which
Sir,
enzymes will be required?
sir, RNA polymerase.
RNA polymerase. And what is transcription? What happens in transcription?
सर डीएनए से आरएनए बनता है.
And how does it happen? RNA polymerase does binds to where?
सब प्रोमोटर पे बाइंड करता है एंड देन
Yes. So guys, if you want to do transcription in a test tube in a lab, all you need is one DNA which has promoter on it, okay? And then you have to add RNA polymerase which will bind to the promoter, and you also have to give something. What else is there? कौन से building blocks दोगे हर्षिता?
आरएनटीपीस.
हाँ, RNTPs, not DNTPs, not DNA वाले, RNA वाले, ribonucleotides. Okay, good. That's nice. So you can make RNA from DNA, and then that RNA you can use, random RNA you can use for finding a riboswitch. So this is a huge, almost PhD problem. You cannot do it in a small project, but I'll try to explain how we make synthetic DNA switch for a ligand for any target that we want. Now let us focus on this slide, and इसी slide को आज समझाऊंगा मैं, and then we'll finish for the day, because next class में मैं आपको offline next process, next step इसका पढ़ाऊंगा, and then riboswitch topic will be over. Now you see how to select a synthetic DNA switch. So synthetic DNA switch you can make, but remember this is not double stranded DNA. This is a single stranded DNA only. Now at the top, so some students have to help me because I cannot show you using this cursor. So you guys have to help me. So what is written on the top? Akshat Verma? Akshat, are you there?
Hello, yes, sir.
Akshat, what is written on the top of this slide?
Sir, how to select a DNA switch tailor mode?
Tailor mode नहीं, tailor made.
Sorry, tailor made. सच चश्मा नहीं लगा है.
Okay. या उसके उसके और उसके नीचे क्या लिखा हुआ है? वो ब्लू कलर की एक बॉल है, उसके लेफ्ट में कुछ लिखा है.
Incubation.
अरे लेफ्ट में मेरे भाई.
Library, sir.
हाँ, लाइब्रेरी. सो वो लाइब्रेरी में आप देख रहे हो छोटे छोटे वर्म्स लाइक स्ट्रैंड्स ऑफ कलरफुल स्ट्रैंड्स आर देयर. So those
sir.
colorful strands, they represent a random library. And what do you mean by random library? It means that you have say like 40 or 50 nucleotide long sequence, but it is randomized. That means
Yes, sir.
जैसे आपको मैं पिछली स्लाइड में दिखा रहा था कि अगर 4 सीक्वेंस रैंडम है, so you have 1034 combinations. So this way if you have 40, you can always say 4 to the power 40. That many different sequences, random sequences you have. Okay. Now what do you do with it? ये जो random sequences हैं, you are hoping that some of them can bind to your target. Now at the right side above incubation, what do you see? Akshat?
Yes, sir. Above incubation, sir, वहाँ से कोई सर वहाँ से फिर हमने शायद इसको सेपरेट करने के लिए और इसमें से सेलेक्ट करने के लिए कुछ एंजाइम डाला है जिससे एक पर्टिकुलर रैंडम में से एक पर्टिकुलर सेट ऑफ डीएनए हमारे साथ बाइंड हो जाएंगे और बाकी के अलग निकल जाएंगे.
अच्छा, नाउ लुक एट द बॉटम अक्षत. बॉटम में
Yes.
इनसेट में कुछ कुछ लिखा हुआ है, एक्सप्लेनेशन है इनका.
यस सर, टारगेट मॉलिक्यूल हमने एक डाला जिसमें
Right.
हमारे जो मतलब जो सेलेक्टेड डीएनएज हैं उससे बाउंड हो जाएंगे और बाकी सारे अनबाउंडेड रह जाएंगे.
हाँ, okay. Now let me modify what Akshat said a little bit, but I'm very happy that he could understand by the diagram. Now, when you throw in the target molecule, it is expected that out of 10 to power 14 different sequences that you have taken in the library, some of them will bind. As Akshat was saying, he was saying like our selected molecule will bind. No, abhi wo random hai. We are selecting them. We cannot still say that they are selected. Okay. So now something is bound, and almost 99.9% of the sequences will not bind because they are not there to bind to the molecule. Now you have to find a way of now separating it. Silpesh, are you there?
Yes, sir.
Silpesh, next step batao. You have mixed your target with random sequences of DNA. Now what is the next step?
So now the selected will bind with the target and the left out will get separated. So
Yes, so you separate with the unbound ones. Okay, fine. So
Yes, sir.
you can have filter technologies also. You can have a sieve where unbound DNA will go down and the on top will stay the target with the bound sequences. So you can do that. Okay. Now next step is Silpesh. Next step क्या है फिर bottom में क्या लिखा है?
Recovery of bound aptamers.
Yes, recovery of bound sequences or aptamers. Now that they are binding, we can call them putative aptamers. So you take them out and then you make a double-stranded DNA out of this single-stranded. So what is the way by which we can make double-stranded from single-stranded? Anishka? Anishka
Yes, sir.
जी,
Yes, sir.
अनिष्का बताओ सिंगल स्टैंडर्ड से डबल स्टैंडर्ड कैसे बनाओगे?
सर प्राइमर्स डाल देंगे,
और फिर
फिर PCR कर देंगे.
okay, yes, that's good. So you do जहाँ amplification लिखा है, you do a PCR. Okay. Now after PCR, after doing the PCR, then you make it single stranded again. So making single stranded, I don't think anyone could answer that. So I'm going to teach you separately how to make a double stranded DNA as single stranded. But if you use your basic knowledge of biochemistry, you can tell me, Anishka,
Yes, sir.
how can you make double stranded DNA as single stranded?
और हेलिकेज एंजाइम डाल देंगे सर.
वेरी नाइस. और सिंपल केमिस्ट्री का नॉलेज लगाओगे कुछ?
फॉस्फोडाइस्टर बॉन्ड ब्रेक करेंगे.
कैसे करोगे? फॉस्फोडाइस्टर नहीं, और बताओ. आयुष पटेजा साहब.
So we have to break the hydrogen bonds between the bases.
Two strands of DNA,
So
you can break the hydrogen bonds. So how do you break the hydrogen bonds? हाइड्रोजन कोई भी बॉन्ड को तोड़ना है तो क्या लगेगा? एनर्जी, है ना?
heating.
Right. So you can heat the DNA and the hydrogen bonds will be broken. It becomes single stranded, isn't it? But the problem is the moment you remove the heat, you remove the tube from that heating instrument, they will anneal back because bonds again will be formed, right? है ना? But there are ways by which you can keep the DNA single stranded form, and I'll teach you when I'm teaching you physically how to make it single stranded. Okay, now let us go to the next step. Ayush, I still need you to help me.
Yes, sir.
So what does it say? I mean, let's not get out of the loop, but in the loop, what does it say?
Sir, after we have amplified the and made the double-stranded DNA, after that it says digestion and then transcription.
Or transcription.
And
now the thing is, yeah. अभी लूप में
Only-
नहीं, लूप में ही रहना है. अभी लूप में ही रहना है, इसीलिए मैं कह रहा बाहर नहीं जाना.
okay.
Right? We don't want to go to the left. Cloning sequencing पे अभी नहीं जाएंगे. So guys, this is now one round of selection, one cycle of selection we have done. Okay. So then same way now it is repeated time and again. So this time when you go to the library, you don't take random sequences. You take this PCR amplified one which you have made single stranded, take that sequence, again bind the target, again separate them, again recover the bound sequences, again amplify them and take it to the next round. Now this way, when we are doing this selection in the lab, when we are selecting for these DNA switches in the lab, we are doing it at least 10 to 15 cycles. Okay, so that's why I was saying like making the aptamer sequence for a target, it is almost a PhD job actually. So you take at least 6 months to 1 year because you get failures also. Lot of time, there will be contamination, lot of issues will be there, but you can do it. And in our lab, my PhD, at least 3 PhD students have done this kind of DNA switch selection, and the results have been very encouraging. Now, so for this one, now you keep on doing, and keep on doing. And now, Ria, I need help from you.
Yes,
Ria?
sir.
Okay, now let us say we have done this cycle 10 times. So this is called as selects. So this cycle goes minimum of 5 times and maximum 16 to 18 times. Okay. Now this selects, now you go to the left side of this. Now what does it say?
So cloning and sequencing.
Hanji, cloning and sequencing. Now because now you want to know what are those selected sequences, what are those DNA switches that we have selected. Okay.
Yes, sir.
So what you have to do is now you have to clone and sequence. So it's hardcore molecular biology now in play, that you use some cloning vectors, and those cloning vectors you use for cloning these sequences, and then you get the sequencing done. Okay.
Yes.
Now when you get, yeah, so when you get this, now thank you, Ria. So now when you get these sequences done, after that, then you have individual sequences. Now you have like 10 different kinds of sequences which are binding to this target, but then you want to select the ones which are best binders, which bind very strongly and which can be used for either gene regulation purposes or biosensing purposes. Okay. Now, so this much for today's class, but I want all of you, this slide is still here. I want all of you to draw the same diagram and upload it on the WhatsApp group within next 15 minutes. Please do that. Is it clear? अरे बच्चों समझ में आ गया?
Yes, sir.
So please start drawing it and upload it on the WhatsApp group right away. अभी 3 बजने में 5 मिनट है. I want by 3:10 all of you to upload this. Title सेम ही रखना है, how to select a DNA switch. Rather you can make how to select a synthetic DNA switch. Okay. अभी ड्रॉ करो, लीव नहीं करना है मीटिंग को. ड्रॉ करके अपलोड करके देन ओनली यू विल लीव द मीटिंग. तब तक मैं देख लेता हूं कौन, अदिति, तब तक मैं अटेंडेंस मार्क कर रहा हूं, आप लोग अपना करो.